So as Steve mentioned, I'll be talking about some perspectives that I have on where total body PET could have a particular impact in clinical research and drug development, which in view of the marked advances which have been happening over the last 5 years in particular in new radiopharmaceuticals and theranostics, really provides an opportunity for Australia to provide leadership and for total body PET to be a key part of this. And these are my disclosures for funding, patents, advisory boards, and board memberships. So what are the key issues that we need to be thinking about in clinical trials, and in particular in early stage drug development, and what can PET do in assisting in the development of new imaging tools and therapeutics? Well, as we all know, and it'll be discussed also by Nicholas later in the session today, there are some properties of total body PET which really stand out for performing early phase clinical trials. Reduced time of acquisition is key. Many of the patients that we put onto clinical trials, particularly for oncology trials, are patients with advanced disease. And if we're going to be using traces where we want to do long scan timeframes, and particularly if we're using long-lived PET traces such as zirconium-89, copper-64, or I-124, and we want to image it for multiple days, reducing the time that the patient's on the scanner is critical. If you're doing a zirconium-labeled antibody, for example, when you're getting out to 5 to 7 days post-infusion, the patient may need to be on a standard digital PET scanner bed for at least 30 or 40 minutes. And so, of course, with total body PET, the reduction in time of acquisition is incredibly important and cannot be underestimated for this type of patient study. And reduced dose, I think, is also very important. For a number of the studies that we do, and I'll show some examples where we have normal volunteers, to be able to reduce the dose that's being administered to the patient or to the volunteer is very important. And clearly there's a trade-off between image quality versus the dose which is given, but that's also a critical factor as well. And of course, we know about the advantages of the increased sensitivity for the total body PET units. We'll be hearing more about that later in the session today. And the ability to pick up lesions more clearly compared to conventional PET scanners is well established. I would recommend this particular article to those of you who are interested in drug development and the role of total body PET. And I draw your attention to where PET, and in particular total body PET, can play a particular role in the development of any new drug. Certainly when you're doing first-in-human studies, to understand what the biodistribution and pharmacokinetics are of the drug can be very important. This can be for a whole host of different small molecules or recombinant proteins or antibodies, where it's possible that the formulation of the particular drug and the targets that it's thought that they're going to, and the deliverable drug concentration to the tissues or tumor that you're aiming for, needs to be established up front. Otherwise, you'll go to large phase 2 trials, don't get a signal of efficacy. You've spent 4 to 5 years and $100 million without being able to make decisions early in the development course. And I'll show some examples of that. And clearly, if you're looking at a diagnostic or a therapeutic involving a radioisotope, it's clear that doing dosimetry is critical. And that also can be markedly facilitated by total body PET as well. So when you're looking at the first-in-human safety and pharmacokinetics, the ADME process, that really is enormously facilitated. And of course, in later studies, it can help with screening and the time that patients are on scanners as well. And again, when looking at the ability to perform dosimetry, having that data in the very first in-human or phase 1 trial program can be enormously beneficial. The other option, of course, and this has been discussed also in prior presentations during this total body PET series, is the potential ability to give multiple traces at a single sitting because of the very low dose that can be given with an initial tracer, followed by the larger dose after an appropriate period of time waiting for the second. What this means is that you can therefore potentially look at a range of different imaging modalities within the same patient on the same day, again, facilitating the experience for patients who are usually not that well all the time, and makes the protocol and clinical trial run much smoother. Depending on what the dosimetric analysis is and whether the kinetics of uptake and the perfusion of the tumour is relevant, of course, the dynamic acquisition and analysis and dosimetry that can be obtained, including parametric images, FDG delivery rate, and blood delivery, can also be extracted very, very well as well. So I'm going to give some examples of where total body PET can play a potential role in drug development, highlighting a couple of programs that we've been involved with, where if we'd had a total body unit, it would have made the translation just so much easier. And we know that in the context of oncology, there are a host of new targets, new therapeutics and imaging tools that are being developed. And the theranostic space is really exploding at this time. So in my own laboratory, we actually, quite a few years ago, developed an antibody which bound to the epidermal growth factor receptor, or EGFR, which we felt was potentially tumor specific. And this was something that, even though we developed immunohistochemical tests to show that the antibody didn't seem to bind to skin or gut or liver, none of the preclinical studies would convince everybody. And so in the first in-human study that we undertook, we actually performed imaging of the antibody itself to confirm the biodistribution. And of course, we did preclinical studies as well to demonstrate the specificity and saturability, and also its impact on the signaling pathway, but also the internalization properties, which led it to be able to be used for drug or for other isotope payload deliveries. And these are the types of studies which can really determine if a new drug will go ahead or not. And what you can see in the whole body image is essentially normal blood pool, a little bit of liver uptake, but nothing like what you would expect if you were binding to wild type antibody. And at very, very low doses, we were able to see uptake in tumor, whereas with antibodies to wild type EGFR, you'd have to give up to 6 to 8 mg per kilo loading doses to saturate normal tissue. So these types of imaging studies can be incredibly important. In this particular trial, we even saw some slight responses at the highest dose level. And this molecule has gone on to subsequent phase 1, phase 2, and even phase 3 trials with AbbVie. Now, FAPI's clearly become one of the most popular and most published and presented molecules to target fibroblast activation protein. And many of you would be familiar with this wonderful illustration of the range of different tumor types which can be targeted with FAPI-based molecules, remembering that FAPI is really a class of ligand which can bind to FAP. And there are other ways you can bind to FAP with smaller molecules or large proteins as well. But to actually interrogate FAP and what its distribution is, either in cancer or inflammation or fibrosis, it's very clear that we need to be able to image multiple patient populations, not just with cancer, but with other fibrotic conditions as well, even with myocardial infarction. And you may say, well, what's the role of total body PET in this context? Well, clearly it will facilitate the conduct of the clinical trials in a very, very expeditious way. But I thought that I would just show an example of the number of companies that are actually developing new FAP molecules at the moment. So Novartis has its own, and it's combining with 3B Pharmaceuticals and took over from Clovis for their molecules. Point Biopharma has been taken over by Lilly, Sophie by GE Healthcare, Lantheus have their own, Yantai, Philogen, and Roche are all just examples of companies, and there is probably a dozen other smaller companies that are developing FAP-based molecules. So I do think that for the first in human studies, even with FAP, there's opportunities for us to be able to utilize our network and to explore total body PET in this context. Another example is targeting the tumor microenvironment. This is a particular molecule FA3, for which we have the crystal structure here, which is expressed in the vasculature and stroma of tumors, as well as myeloid-derived suppressor cells. And in order to demonstrate that the microenvironment could be targeted with this particular therapeutic approach, we conducted a trial with zirconium-89 labeled ifabituzumab and showed for the first time targeting of the microenvironment in glioblastoma. And again, this has accelerated the development of this particular molecule as an ADC and also for other payload deliveries with alpha and beta particles, illustrating how early phased imaging with PET and with a total body system can facilitate this type of development further. Now, imaging of immune checkpoints has also emerged as a very important way of assessing not just approved therapeutics, but also some of the newer bispecific and immune target agents. This is an example of pembrolizumab and its uptake and demonstration that the uptake of this particular zirconium pembrolizumab probe was correlated with therapeutic response. And as part of the NIF network, we have developed methodology for automated labeling of durvalumab with zirconium-89. This has been extended to a series of studies that are single site. This is an example of imaging of lymphoma with zirconium durvalumab. So you can see in the right axilla, PD-L1 expression imaged by durvalumab and assessment of whether that's predictive of response in patients who are getting multiple therapies. And there are a range of different multi-center trials which are now commencing at the moment throughout the NIF network, which I think can also be facilitated by performing total body PET and linking to what's been announced as a total body PET platform across the country, which we hope to be seeing over the next few years. We can also image T cells. This is an example of the trafficking of T cells into the left axilla in a patient with lymphoma with a mini-body labeled with zirconium-89. And another example of where immune-based imaging can be facilitated. And in an example from Professor Simon Cherry, which was shown at the opening of the total body PET unit at Royal North Shore Hospital and University of Sydney, the imaging of T cells in COVID recovery with total body PET really provides remarkable evidence of exactly what's happening with viral load. We've performed studies with approaches to image HIV, restricted because of the dosing to look at normal volunteers as well as in patients. We actually had to use copper-64. And you can probably appreciate that you could only image copper-64 up to 48 hours after injection with fairly low count rates. This is an example of where having total body PET with higher sensitivity, we would be able to go out to even 5, 6, 7 days and to really interrogate what the ability of the probe, which targets the HIV protein itself, can actually show in this population. We've been working for a number of years with a range of different biotech and pharmas looking at novel immunotherapy approaches. This is a bispecific against PD-L1 and TGF-beta, which reduces the immune suppressive microenvironment. And we undertook work in preclinical studies to be able to demonstrate the binding to tumor and both PD-L1 and TGF-beta, and took this into a clinical trial in patients with non-small cell lung cancer. And what you can probably appreciate just in the right-hand side of the image is that the uptake within this mediastinal mass just here is in fact very low. And we were able to show that the drug deliverable concentration was lower than they were expecting, which then coincided with their phase 2 study results showing lack of response, clearly indicating with some of the newer therapeutic molecules being able to perform whole body imaging at multiple time points is essential if we're going to be able to stratify those drugs which need to continue or to halt them earlier. We know that if we're looking at cancer therapy, a range of different approaches can be used to enhance therapeutic effect. This can be either through DNA repair inhibitors, increasing the receptor expression, for example, with the androgen receptor, with enzalutamide. We can image, as I've shown you, the immune function. But there are trials which are happening in Australia to look at ways in which we can upregulate target and identify if redifferentiation can have an impact. And an example of that, and thanks to Dale Bailey, who's provided this example from the iFirst study, which is being conducted in patients with radioiodine refractory thyroid cancer, and where we're giving redifferentiation therapy to upregulate sodium iodide syn-porter expression, and then to image to see if we were able to give more than 20 gray calculated with I-131 as subsequent treatment. And you can see here a 40-minute scan on the left-hand side with the standard PET camera compared to a 12-minute scan that was performed on the total body system, which is at North Shore and University of Sydney, clearly illustrating the ability of total body PET to facilitate some of these early stage validation studies for radionuclide therapy. I would emphasize that the importance of developing networks is something that will contribute to the way in which total body PET will engage with the community and also contribute to drug development. And with the National Imaging Facility proposing to develop a total body PET network, that means that we will be able to see this extending throughout the country, and we look forward to seeing how that will progress. I would mention that the UK has started to develop a program for total body PET network for clinical trials. Something called the NPIP, or National PET Imaging Platform, has been announced. I don't know that there's one in the US at the moment, but certainly it highlights the importance of developing these networks, and we'll see more of this in the future. It's not just looking at cancer or looking at just the whole body, for which total body systems can be very helpful. In neuroscience, where we want to be able to image patients and volunteers on multiple occasions, and we want to be able to do this in an effective way, you can use the systems in this regard. These are just some examples, and thanks to Dale Bailey as well, of FDG, DAT scan, and also amyloid scans, which can be evaluated. And with amyloid, it can be used both for brain amyloid as well as for total body assessment for non-brain amyloid accumulation, such as in the heart or in other organs. And there's even implications for inflammatory and infectious disease. This is just one example of looking at different types of arthritis, the patterns of distribution, the intensity of uptake, and for sequential studies with reduced dose, reduced time on scanner, I think will be increasingly important as we develop trials looking at interventions and where PET is used for assessment. So I think that we've got some terrific opportunities to take discoveries into the clinic, both academic and with industry, and I hope that the presentation I've given provides some context to that. Thank you.